breakdown is photorespiration, an energy-wasting process. The energy and reducing power used to produce the two reduced carbons of phosphoglycolate are completely lost. Photorespiration is extremely exergonic but is not used to provide power to endergonic reactions. Because phosphoglycolate is toxic, photorespiration protects the plant whenever Rub-P carboxylase picks up oxygen. However, it is an expensive defense because up to 30% of all ATP and NADPH produced by the chloroplast can be immediately lost by photorespiration. Figure 10.26 If Rub-P carboxylase puts oxygen onto Rub-P, one of the products is phosphoglycolate, which is transported to a peroxisome and broken down during photorespiration. All of the energy that was present in the phosphoglycolate is wasted. Mitochondria are also involved. Apparently, Rub-P carboxylase cannot be significantly improved. At the time when Rub-P carboxylase originated by evolution, there was no free oxygen at all in the atmosphere, and carbon dioxide concentrations were very high. Rub-P carboxylase was well adapted to that environment. The first type of photosynthesis to evolve began to remove carbon dioxide from the air, but it did not produce any oxygen, so conditions were still appropriate for Rub-P carboxylase. Photosynthesis that produces oxygen originated in cyanobacteria about 2.8 billion years ago, and ever since, Earth's atmosphere has slowly changed such that Rub-P carboxylase does not function well, oxygen has increased and carbon dioxide has decreased. Massive volcanic eruptions occasionally increase the concentration of carbon dioxide in the air, but molecule by molecule, Rub-P carboxylase pulls it out, altering its environment to its own detriment. Rub-P carboxylase has existed for billions of years, and virtually no structural mutations have survived. They must not have produced superior versions of the enzyme. An alternative is to improve the working conditions of Rub-P carboxylase. Rub-P carboxylase should be compartmentalized in a site where carbon dioxide concentration is high and oxygen concentration is low. This has evolved in some plant groups and is known as C metabolism or C photosynthesis. Basically, C metabolism is a mechanism by which carbon dioxide is absorbed, transported through, and concentrated in a leaf, whereas oxygen is kept away from Rub-P carboxylase. C metabolism occurs in leaves with Krantz anatomy. In such leaves, mesophyll is not distributed as palisade and spongy parenchyma, rather, each vascular bundle has a prominent chlorophyllous sheath of cells, and around the sheath are mesophyll cells. Mesophyll cells contain the enzyme PEP carboxylase, which has a very high affinity for carbon dioxide. Unlike Rub-P carboxylase, as the carbon dioxide concentration drops lower and lower, PEP carboxylase continues binding to it rapidly and firmly. Carbon dioxide concentrations inside the leaf are kept very low, and carbon dioxide diffuses inward rapidly whenever stomata are open. The ratio of water lost to carbon dioxide absorbed is favorably low. Also, PEP carboxylase has a high specificity for carbon dioxide, it never picks up oxygen. PEP carboxylase is an ideal enzyme except that it does not perform the critical reaction that results in 3-phosphoglycerate. Despite its shortcomings, Rub-P carboxylase is still the only enzyme that carries out the necessary reaction. Figure 10.27 In leaves with Krantz anatomy, the bundle sheath around all veins is prominent and rich in chloroplasts. The bundle sheath chloroplasts are located as closely as possible to the vascular tissues. The mesophyll cells are arranged in a sheath around the bundle sheath. Knowing the reactions of C metabolism is not sufficient for understanding it, only by realizing that some reactions occur in separate compartments does it become logical. PEP carboxylase adds carbon dioxide to PEP, producing oxaloacetate, which has 4 carbons, hence the name C metabolism. Oxaloacetate is reduced to malate by a molecule of NADPH, and further reactions may occur, depending on the species. Malate from throughout the mesophyll moves into the bundle sheath and breaks down into pyruvate by releasing carbon dioxide. This reaction is powerful enough to drive the formation of a new molecule of NADPH, so the process results in the transport by malate of both carbon dioxide and reducing power. Because all of the malate from a large volume of mesophyll decarboxylates in the small volume of the bundle sheath, carbon dioxide concentration in the sheath is very high. Typically, the concentration of carbon dioxide in bundle sheath cells is 10 times higher than it is in air. Also, because NADPH is synthesized by this unusual method in bundle sheath cells, the bundle sheath chloroplasts primarily carry out cyclic electron transport, pumping protons and making ATP. Without non-cyclic electron transport, there is no breakdown of water or production of oxygen. Rub-P carboxylase, located exclusively in the bundle sheath chloroplasts, is in ideal conditions, a high carbon dioxide concentration with low or no oxygen, and carries out efficient C-stromal reactions with low production of phosphoglycolate. C plants have little photorespiration. Like any other carrier, malate must be shuttled back to its recharge site, the mesophyll. In the bundle sheath it is converted to pyruvate by the release of carbon dioxide. Pyruvate moves back to the mesophyll and receives a phosphate group from ATP, which converts it to PEP. The selective advantage of C metabolism depends on the environment. Approximately 50 million years ago, the concentration of carbon dioxide in the air dropped to a critical level and global temperatures cooled. 20 million years later, Earth entered the Pleistocene Ice Age and ever since has been dry and low on carbon dioxide. Conditions such as these favor the increased water use efficiency of C metabolism, and it began to evolve independently many times. At present it occurs in 19 separate plant families. Photorespiration increases with temperature, so it is more of a problem in hot climates. Under warm, dry conditions, C metabolism has a strong selective advantage over C metabolism. Much less water is lost during carbon dioxide absorption. Also, abundant light is available to generate the extra ATP needed to convert pyruvate to PEP. The ATP used to make PEP means that C metabolism is not free. Under cool conditions, photorespiration may be slow enough that it loses less energy than C metabolism. Also, many cool habitats are also moist, so water conservation by stomatal closure is not as critical. The critical temperature above which C metabolism is more advantageous selectively than C metabolism varies among species but averages around 25 degrees Celsius. In general, the optimum temperature range for C photosynthesis is 20-25 degrees Celsius, and for C photosynthesis it is 25-30 degrees Celsius. Table 10, 6 types of carbon dioxide processing. Table 10, 7 plant families having C species. C metabolism and Kranz anatomy have evolved several times. Most C species are monocots of hot climates such as corn, sugarcane, sorghum, and several other grasses, but a considerable number of eudicots, also from warm, dry regions, are also C species. Table 10-7. Crassylation acid metabolism. Crassylation acid metabolism is a second metabolic adaptation that improves conservation of water while permitting photosynthesis. It is so named because it was first discovered in those members of the family Crassulaceae that have succulent leaves. The metabolism is almost identical to that in C plants. PEP is carboxylate, forming oxaloacetate, which is then reduced to malate or other acids. These acids are not transported but simply accumulate, in effect storing carbon dioxide. This occurs at night. These plants differ from C and C plants in that their stomata are closed during the hottest periods and open only at night when it is cool. Coolness reduces transpiration, and usually air is calmer at night, therefore, water molecules near stomata are not blown away immediately and may diffuse back into the plant. Opening stomata at night is effective for conserving water, but the lack of light energy creates a problem for photosynthesis. A plant cannot store ATP and NADPH during the day for use at night. They are not stable enough and each cell has too little Hence, carbon dioxide is stored on acids until daytime, when stomata close and the malate or other acids break down, releasing carbon dioxide for C metabolism. The released carbon dioxide cannot escape because stomata close at dawn. Figure 10.28 Sempervivum arachnoideum, a member of the Crassulaceae. Like many desert succulents, it has CAM metabolism as well as other adaptations that conserve water. This barrel cactus is a CAM plant. Photosynthetic cells of CAM plants have large vacuoles, which permits the accumulation of C acids. These are cortex cells, arranged in palisades, similar to leaf palisade mesophyll. Crassulation acid metabolism is not particularly efficient. The total amount of carbon dioxide is so small that it may be entirely used in C metabolism after just a few hours of sunlight. Furthermore, ruby P carboxylase is protected from photorespiration only in the morning when internal carbon dioxide levels are high. Later in the day, photorespiration may be high, but the carbon dioxide released by the breakdown of phosphoglycolate is trapped and refixed. Alternatives. Box 10-2 Photosynthesis in bacteria and cyanobacteria. A process as complex as photosynthesis had to evolve by numerous steps, and many early types of photosynthesis are present in bacteria and cyanobacteria. By examining them, we can understand plant photosynthesis more fully. Cyanobacterial photosynthesis is the most similar to that of plants. This is not surprising because chloroplasts arose as endosymbiotic cyanobacteria living inside early eukaryotic cells. Cyanobacterial light reactions are almost identical to those in chloroplasts, having chlorophyll but lacking chlorophyll B. Their accessory pigments are phycobilins, open-chain tetrapyrrole rings, which act like carotenoids by absorbing wavelengths that chlorophyll cannot and then transferring the energy to chlorophyll and activating an electron. One class of phycobilins, phycocyanin, absorbs most strongly at approximately 620 to 640 nanometers and is blue. The other class, phycoerythrin, is red because it absorbs maximally at 550 nanometers. Both occur bound to proteins, and these aggregate into small nodules visible by electron microscopy. Most other aspects of photosynthesis in cyanobacteria are identical to those of plants. Electrons are taken from water, pass through photosystem 2 to psi, and are then used to reduce NADP to NADPH. Oxygen is liberated as a waste product, as in plants, therefore, both have oxygenic photosynthesis. Cyanobacteria do not have chloroplasts, but they do have extensive sheets of enfolded plasma membrane that contain pigments and electron carriers. The folded membrane forms accumulation spaces for protons and the generation of a chemiosmotic gradient. Figure B10, 2a a typical phycobilin, containing 4 pyrrole groups, a tetrapyrrole. The two ends of the pyrrole are not attached to each other as they are in chlorophyll. Purple bacteria and green bacteria do not contain chlorophyll either A or B, but instead have bacteria chlorophylls. Like chlorophyll, these are closed tetrapyrroles with a long tail, but they have certain side groups that the chlorophylls lack. Carotenoid accessory pigments are present, as in chloroplasts. PSI operates, but PS2 is not present to put electrons back onto bacteria chlorophyll. Instead, the original electron comes back through a series of carriers, so electron flow is cyclic, similar to that in plant photosynthesis. One carrier is quinone, thus, protons are pumped across the photosynthetic membranes, creating a strong chemiosmotic gradient that causes ADP to be generated, just as in eukaryotic chloroplasts. Because there is no PS2, oxygen is not formed. This is anoxygenic photosynthesis. The photosynthetic apparatus of purple bacteria consists of extensive arrays of membranes connected to the plasma membrane. In green bacteria, photosynthetic membranes are cylindrical vesicles that occur in clusters surrounded by another membrane. The enclosing membrane does not look like a typical bilayered membrane in electron micrographs and is probably different physiologically as well. Notice that bacterial photosynthesis is basically just light-powered proton pumping. It is extremely simple and not very effective. Bacteria chlorophyll does not capture enough light energy to activate an electron strongly enough for it to move onto an ADP. Instead, electrons must cycle repeatedly, gradually building up enough of a chemiosmotic gradient to finally produce an ADPH. In contrast, chlorophyll captures enough energy to reduce an ADP in just 2 steps. Also, plants and cyanobacteria obtain all of the electrons they need from water, which is always present wherever organisms are alive. Purple and green bacteria recycle electrons repeatedly as they pump protons, but the electrons used to reduce an ADP must come from other substances, such as sulfur and hydrogen, which are not always reliably present. Figure B10, 2b bacteria chlorophyll is similar to chlorophyll A, differing only at the sites indicated by color. Is this similarity analogous, the product of 2 independent lines of evolution, or homologous, the genes for one pigment having evolved from those for the other? These 2 molecules are not sufficient evidence to judge, but we are almost certain that all photosynthetic systems are closely related evolutionarily. Although plant photosynthesis is complex, think about it in terms of being composed of several parts that originated as simple pumps, cyclic electron flows, and chemiosmotic synthetic pathways. Each could originate as a simple metabolic function that then evolved to become more sophisticated. Gradually, one part was added to another, then another, and finally, chlorophyll-based oxygenic photosynthesis resulted. Figure B10, 2c electrons flow cyclically from activated bacteria chlorophyll A, pumping protons with quinone carriers. This establishes a chemiosmotic gradient that generates ATP. BPH equals bacteriophyophytin. Table 10, 8 plant families having CAM species. Crassylation acid metabolism is selectively advantageous in a hot, very dry climate where survival rather than rapid growth is most important. In these habitats, unaided C. metabolism is so wasteful of water that sea plants cannot survive. In the most arid regions, sea plants barely get by, growing in slightly less stressful microhabitats such as near temporary streams and ponds, or growing and flowering quickly during the cool, moist months of winter and spring and then dying in the summer and surviving only as seeds. Under such conditions, CAM plants have a selective advantage. They may not grow luxuriantly, but they do grow and reproduce more successfully than C and C plants. Under milder, moister conditions, crassylation acid metabolism is not selectively advantageous. Water conservation is less of a benefit, and the limited capacity to absorb and store carbon dioxide is a distinct disadvantage. C and C plants photosynthesize all day, whereas CAM plants may stop before noon. Crassylation acid metabolism has evolved several times and is also present in the cactus family, many orchids, bromeliads, lilies, and euphorbias. All have other metabolic and structural adaptations for water conservation, such as succulent bodies filled with water-storing parenchyma and covered by a tough epidermis with a thick cuticle and wax layer. At the next level. 1. Photosynthesis and carbon sequestration. Box 10-1 is an introduction to the interplay between greenhouse gases, photosynthesis, and global warming. An internet search on this will lead you to many sites, some written by crackpots, so be careful to use your judgment as to whether a site is reliable. One important topic for you to focus on is carbon sequestration, removing carbon dioxide from the atmosphere and locking it away as wood, calcium carbonate seashells, or as carbon dioxide pumped into empty oil wells. Another important topic is whether photosynthesis will automatically speed up as levels of carbon dioxide increase in the air. After all, the current level of carbon dioxide is one of the lowest that has ever occurred, and it could be that plants are now growing more slowly than they ever did in the past. But we are not certain and research is in progress. 2. Photosynthesis and biofuels. Many people suggest that we should cultivate plants such as corn and sugar cane and then ferment the corn and sugar to alcohol to use as a substitute for petroleum. Fuels produced by photosynthesis are biofuels. The problem is that the production of these crops requires gasoline and diesel for tractors to plow, plant, till, harvest, haul, and refine the corn and sugar. If we put in more energy than we get out, we are not only wasting our time, we are making the situation worse. Researching and thinking about biofuels is an important topic by which you can take your thinking to the next level. 3. Chloroplast structure and reaction centers. Our understanding of the biochemistry and biophysics of photosynthesis is expanding rapidly. Advanced textbooks in plant physiology can give you an orderly explanation of photosynthesis, and with the information you have in this chapter, you should have a good foundation. A textbook or review article must be new. Progress is so rapid that a 10-year-old book on photosynthesis is out of date. Light harvesting complexes are especially important. 4. Global warming. Convincing proof exists that global warming is being caused by us, by our burning fossil fuels. It is called anthropogenic global warming. An important step for you to think about is whether global warming is good or bad. We are in an interglacial period of an ice age. Earth has occasionally been colder than it is right now, but more often it has been much warmer and with much more carbon dioxide in the atmosphere. What will be the effect of warmer temperatures? Some species will go extinct, but new species may come into existence and others will flourish. Remember, the scientific method can provide us with facts about this, but you must use your own ethical standards to decide if it is good or bad. Summary. 1. All physical systems have a tendency to become disordered, to increase in entropy. Living organisms have a high degree of order and regularity, maintained by the input of energy, the ultimate source of which is the sun. 2. All photosynthetic plants are autotrophs, but some parasitic plants and many plant tissues are heterotrophic. 3. When electrons are passed from one atom to another, the recipient becomes reduced, and the donor becomes oxidized. Most of a plant's raw materials are highly oxidized and must be reduced as they are assimilated. 4. Photosynthetic pigments respond to light quanta that have just enough energy to boost electrons 1 or 2 energy levels. 5. Accessory pigments have absorption spectra different from that of chlorophyll A, and they therefore absorb different wavelengths and transfer energy to chlorophyll A. 6. Photosystems 1 and 2 work together, transferring electrons from water to NADPH. The electron's energy is boosted twice, once at P680 and again at P700. 7. The light-dependent reactions result in a chemiosmotic gradient. Driven by concentration differences, protons flow from the thylakoid lumen to the stroma through ATP synthetase channels, powering the phosphorylation of ADP to ATP. 8. Cyclic electron transport permits production of extra ATP without synthesis of NADPH or production of free oxygen. Non-cyclic electron transport results in production of both ADP and NADPH, but the amount of ATP is not sufficient for the stroma reactions. 9. All photosynthetic plants use the C-stroma reactions mediated by Ru-BP carboxylase. A molecule of Ru-BP is carboxylate, energized, and reduced, resulting in 2 molecules of 3-phosphoglyceraldehyde. 10. C metabolism is an adjunct to C metabolism, not a replacement. PEP carboxylase, which acts as the initial carboxylating enzyme, has a great affinity for carbon dioxide. 11. Crassylation acid metabolism is similar to C metabolism, except that it accumulates and stores carbon dioxide at night while stomata are open and releases it during the day while stomata are closed. Important terms. Plastoquinones quality of sunlight quantum reaction center reduced compounds reducing power. Ribulose 1,5-bisphosphate, Ru-BP carboxylase stroma-stroma reactions. Substrate-level phosphorylation thylakoid lumen Z scheme. Review questions. 1. What is the meaning of the word entropy? Does the entropy of a plant increase or decrease while it is alive? After it is dead? 2. Name several examples of photoautotrophs and several of heterotrophs. How do photoautotrophs obtain energy? Can a plant be heterotrophic while a seedling and photoautotrophic when older? 3. ATP is an important chemical involved in many of a plant and animal's metabolic reactions. Yet any plant has only a small amount of it. Can you explain this? When ATP enters a reaction and forces it to proceed, what is the ATP converted into? What then happens to that molecule? 4. Name the 3 methods of phosphorylation. 5. What is a reduction reaction? Why does a reduction reaction always occur simultaneously with an oxidation reaction? 6. In organic molecules, we calculate the oxidation state of carbon by assuming that each oxygen has an oxidation state of. Each hydrogen has an oxidation state of. Calculate the oxidation state of carbon in each of the following: CO, CHO, and malic acid. 7. 2 of the following are oxidizing agents and 2 are reducing agents. Which are which? NAD, NADP, NADH, and NADPH. 8. In photosynthesis, what is the ultimate source of electrons? What are the benefits of this molecule in terms of its toxicity and the cost of the plant to obtain it? 9. Describe the absorption spectrum of chlorophyll. Why does it match the action spectrum of photosynthesis? 10. Chlorophyll does not use high-energy quanta. Why not? What would happen to the chlorophyll if it did? It also does not use long-wavelength radiation either. Why not? 11. The most common accessory pigments in land plants are chlorophyll and the algae that live in deep water have other accessory pigments because only light penetrates deeply into water. 12. Name the electron carriers that transport electrons from photosystem II to photosystem I. Which ones contain metal atoms, and which do not? 13. When photosystem I produces NADPH, its reaction center P700 chlorophyll loses electrons. What would happen if photosystem II did not supply new electrons to P700? 14. When electrons are removed from water, protons are liberated. Does this occur in the stroma or inside the thylakoid lumen? Can protons move directly across the membrane? Describe the chemiosmotic mechanism of ATP synthesis in chloroplasts. 15. Is ADP converted to ATP directly by the reaction center chlorophylls? Do the enzymes that synthesize ATP obtain the necessary energy by interacting directly with the reaction center chlorophylls? 16. What chemical is the acceptor of carbon dioxide in the C cycle? What enzyme catalyzes the reaction, and what is the product? 17. Rub-P carboxylase is by no means an ideal enzyme. Describe some of the problems with its active site and its substrate specificity. If we compare the amino acid sequences of this enzyme from many different species, they are almost identical. What is the significance of this uniformity? 18. Which chemicals are useful for energy storage on a short-term basis? Which are for intermediate term and which are for long term? 19. What is the quality of light? How does it differ for plants in deserts, grasslands, and the canopy of a forest versus for plants in the understory? How does it differ for algae that grow near the surface of a lake or ocean versus those that inhabit deep water far below the surface? 20. How is the quantity of light affected by a plant's location relative to the equator or the poles? On one side of a mountain or the other? On one side of a valley or the other? 21. Imagine a leaf in bright light but an atmosphere with no carbon dioxide. Would Rub-P carboxylase be functioning? Would the NADP be in the reduced or oxidized form? 22. Name some of the brightest environments. Describe some protective adaptations that plants may use to shade themselves. 23. An important factor for plants is the amount of water lost for each molecule of carbon dioxide absorbed. How could the plant be harmed if it loses a lot of water for each carbon dioxide molecule, that is, if the ratio is high? Would this be more important for a plant in a rainy habitat or one in a desert? 24. An AC plant, where is PEP carboxylase located? Where is Ru-BP carboxylase located? 25. In a CAM plant, are stomata open during the day or the night? How does this affect the amount of water the plant loses when its stomata are open? 26. As a CAM plant makes and stores acids during the night, how does this affect the plant's acidity? Think about the acidity of your own blood. Do you think it is allowed to vary by any large amount? 27. In habitats where water conservation is not especially necessary, is CAM metabolism more or less advantageous than C or C metabolism? Why? 28. What is global warming? What is the main gas that causes it? What would happen if the Earth's atmosphere had a lower concentration of CO than it has now? What would happen if it had more? 29. What is the Kyoto Protocol? How many countries have signed it? Name one country that has not signed it. What are two substances burned in the United States that produce CO? Which two countries have large populations and may soon surpass the United States in production of greenhouse gases? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated Z-Art. Chapter 11. Energy Metabolism, Respiration. Chapter Opener Image. Plant tissues carry out aerobic respiration just as we animals do, and if submerged in water, they will drown. The roots of these bald cypress trees grow in mud below calm water, a region with almost no oxygen. But the roots send up knees that have thousands of air channels that permit oxygen to diffuse downward from the atmosphere to the roots. As long as the knees protrude above the surface of the water, the roots will have enough oxygen. Outline. Types of respiration. Anaerobic respiration. Aerobic respiration. Heat-generating respiration. Pentose phosphate pathway. Respiration off lipids. Photorespiration. Environmental and internal factors. Temperature. Lack of oxygen. Internal regulation. Total energy yield of respiration. Respiratory quotient. Fermentation of alcoholic beverages. Beer. Wine. Spirits. Warnings. Box 11 to 1 Plants and People, Fungal Respiration. Box 11 to 2 Alternatives, Respiration in Prokaryotes. Box 11 to 3 Plants do things differently, Plants, Babies, and Heat. Box 11 to 4 Plants and People, Respiration. Learning Objectives. After reading this chapter, students will be able to explain what becomes of the excess energy produced by photosynthesis. Compare aerobic and anaerobic respiration. List the 3 parts of aerobic respiration. Identify a benefit of thermogenic respiration. Describe the value of the pentose phosphate pathway. Explain the relationship between temperature and respiratory rate. Describe how aerobic respiration is more efficient than anaerobic respiration. Define the respiratory quotient. Compare the fermentation processes of beer, wine, and spirits. Did you know? A glucose molecule has too much energy to be used in most chemical reactions. Respiration breaks its energy into 36 smaller pieces, each in an ATP molecule. Respiration is one of the most ancient of all metabolisms. Almost all organisms respire by more or less the same metabolic pathways. Aerobic respiration requires oxygen and produces many ATP molecules for each glucose molecule respired. Anaerobic respiration does not require oxygen but produces fewer ATP molecules per glucose molecule. Aerobic exercise activities are actually anaerobic if you run, bike, or exercise so rapidly your blood cannot bring oxygen as quickly as your muscles need it. Concepts. The light-dependent reactions of photosynthesis produce an excess of energy and reducing power, both of which are stored as glucose and starch. An important corollary must be the ability to recover that energy and reduced carbon. Recovery may occur later, when photosynthesis is impossible, such as night or in winter when the plant is leafless. Also, energy recovery may occur at a different site from photosynthetic capture. Glucose may be converted to sucrose, transported to apical meristems, vascular cambia, or any other heterotrophic tissue, then broken down to recover the energy. Respiration is the process that breaks down complex carbon compounds into simpler molecules and simultaneously generates the adenosine triphosphate used to power other metabolic processes. During respiration, carbon is oxidized. Its oxidation state goes from +0 to +4 as electrons are removed by NAD, which is converted to NADH in the process. This is basically the opposite of photosynthesis, in which NADPH carries electrons to carbon, reducing it. The NADH generated by respiration is a good reducing agent, but it is produced in much larger quantities than needed for constructive reduction reactions. Plants use mostly NADPH from photosynthesis for their reductions, not NADH, and most of the compounds that animals and fungi consume are already reduced. Because NADH contains a great amount of energy, it is selectively advantageous for an organism to be able to oxidize it so as to generate even more ATP. Oxidation of NADH to NAD requires transferring electrons from it onto something else. The ideal recipient would be abundant, cheap, and non-toxic after it is used. These are also the three ideal characteristics of the source of electrons in photosynthesis, and the chemical involved in both processes is water. As it turns out, the result of respiration is the reverse of photosynthesis. Electrons are transferred from carbon and carbohydrate by means of reduced NADH, which carries them to an electron transport chain, which in turn deposits them onto oxygen, reducing it. As electrons are added, protons are attracted and incorporated, converting oxygen to water. If this reverse analogy were carried one step further, the electron transport chain would have to give off light, but that would be a complete waste of energy. Instead, it conserves the energy as high-energy phosphate bonding orbitals of ATP. Respiration is also important to the plant because numerous intermediate compounds of its many steps are useful as starting materials for several anabolic pathways. A molecule of glucose, after entering the respiratory pathway but before being broken down to carbon dioxide and water, may be picked up in the form of an intermediate by a non-respiratory enzyme that diverts it into a pathway that produces amino acids, fats, nucleic acids, lignin, and other molecules. Figure 11. When light is available, autotrophic tissues produce more ATP and NADPH than needed for cell metabolism. Glucose is produced, part of which is stored as starch in the leaf and part converted to sucrose and transported to areas that are either completely heterotrophic or are growing more rapidly than their own photosynthesis can support. Sucrose is converted back to glucose and is either respired or used in the construction of cell structures such as cellulose, lignin, amino acids, and nucleic acids. In roots, trunks, fruits, and seeds, some glucose may be polymerized to starch and stored for months or years. Even autotrophic tissues are heterotrophic in the dark, surviving on respiration of stored starch. Figure 11. 2. This hypothetical reaction diagram shows the relative levels of potential energy of the major compounds of photosynthesis and respiration. Energized P700 has the greatest potential energy, most of which is trapped in the formation of ATP and NADPH. When these reduce carbon dioxide and make glucose, much of the energy is conserved. Respiration transfers the energy to ATP and NADH. During other metabolic reactions, the breakdown of ATP and NADH yields large amounts of energy. Depending on the metabolic pathway, some of this energy is retained and some lost. Figure 11.3. It is selectively advantageous for an organism's metabolism to be based on raw materials that are abundant and cheap and produce non-toxic waste products. Mutations that cause an individual to require rare or expensive compounds or those that break down into toxic products are selectively disadvantageous. Table 11.1. Relationships between types of organisms and presence or absence of oxygen. Types of respiration. Cellular respiration falls into two categories, aerobic and anaerobic. Respiration that requires oxygen as the terminal electron acceptor is aerobic respiration. Under certain conditions, oxygen is not available, and an alternative electron acceptor must be used. This is anaerobic respiration, respiration without oxygen, often called fermentation. Because animals and plants must have oxygen for their respiration, they are known as obligate or strict aerobes. At the opposite extreme are certain bacteria called obligate anaerobes, which carry out anaerobic respiration exclusively. Such bacteria are actually killed by oxygen. Many fungi and certain types of tissues in animals and some plants are facultatively aerobic. If oxygen is present, they carry out aerobic respiration, but when oxygen is absent or insufficient, they switch to anaerobic respiration. Although many fungi, especially yeasts, can live indefinitely anaerobically, plant and animal tissues can survive this way for only a short time. They must eventually obtain oxygen and switch back to aerobic respiration or they die. Anaerobic respiration. Glucose is broken down during anaerobic respiration by a metabolic pathway called glycolysis or the Embden-Meyerhof pathway. Glycolysis and gluconeogenesis are essentially the same pathway, with the reactions running in opposite directions, figure 11. Although all intermediates are the same, as are many of the enzymes, the two processes use different enzymes at certain key steps. This allows a cell to regulate the two processes so that one is stopped while the other runs. It would be useless for both pathways to operate simultaneously within a single cell. Figure 11, for glycolysis, also called the Embden-Meyerhof pathway, constitutes the major portion of anaerobic respiration and is also the first part of aerobic respiration. In glycolysis, ATP phosphorylates glucose to glucose 6-phosphate, which is then converted to fructose 6-phosphate. A second molecule of ATP then phosphorylates this to fructose 1,6-bisphosphate, which breaks down into 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. The latter can be converted into a second molecule of 3-phosphoglyceraldehyde, and both can be oxidized to 1,3-diphosphoglycerate. During this oxidation step, electrons are transferred from a carbon of 3-phosphoglyceraldehyde to NAD, converting it to NADH. The 1,3-diphosphoglycerate is energetic enough that an enzyme can transfer one of its phosphate groups onto an ADP, converting it to ADP and changing the 1,3-diphosphoglycerate into 3-phosphoglycerate, a process called substrate-level phosphorylation. The enzyme is phosphoglycerate kinase. The kinases constitute a large group of enzymes that remove phosphate groups from substrates. Phosphorylases are just the opposite, adding phosphates to substrates. Although this is basically a reversal of the stromal reactions, ribulose 1,5-bisphosphate does not occur next as one might expect. Instead, 3-phosphoglycerate is converted first to 2-phosphoglycerate and then to phosphoenolpyruvate, the same metabolite that is the carbon dioxide acceptor in C metabolism and crassylation acid metabolism. PEP is also energetic enough that an enzyme can transfer its phosphate group onto ADP to make ATP. Dephosphorylation causes PEP to become pyruvate. If no oxygen is present, anaerobic respiration has now removed all the energy possible. From each molecule of glucose, 4 ATPs were generated and 2 were consumed, thus, there is a net production of 2 ATPs. Occasionally, the cell can start with glucose 6-phosphate and use 1 less ADP to get started. The ATP generated in anaerobic respiration is used for other metabolic reactions such as protein synthesis, nucleic acid replication, microtubule assembly, and ion transport. Indeed, these metabolic pathways are the reasons for respiration, and the ADP they generate migrates back to the sites of glycolysis and is rephosphorylated to ATP. The reduction of NAD to NADH during glycolysis is a problem. If the cell needs reducing power, the NADH can be used, and it regenerates the NAD necessary to keep glycolysis running. For example, roots absorb nitrates and sulfates that must be reduced, and during the synthesis of fatty acids, large amounts of reducing power are needed. Many of these reductions can be carried out with this extra NADH, but usually a cell does not need as much reducing power as is produced during respiratory ATP production. Consequently, NADH accumulates, all the NAD is consumed, and glycolysis stops for lack of NAD. Without further glycolysis, no ATP would form and death would result. The big problem is converting NADH back to NAD by dumping its electrons onto something. In animal tissues under anaerobic conditions, the electron acceptor is pyruvate, and NADH reacts with it to form lactate, the anion of lactic acid. In plants and fungi, pyruvate is first converted to acetaldehyde, and then NADH reacts with that, forming ethanol. Anaerobic conditions occur in plants and fungi growing in mud beneath stagnant water, especially in swamps and marshes. Rice seeds germinate and grow anaerobically until the shoots reach oxygenated water. Figure 11.5 If a cell needs reducing power, as for the synthesis of fatty acids, those reactions are actually electron-accepting reactions and they regenerate NAD, which diffuses to other parts of the cell, allowing the continued production of ATP by glycolysis. If reducing power is not needed, NAD accumulates as NADH. Without NAD, glycolysis stops. 3-phosphoglyceraldehyde cannot be oxidized to 1,3-diphosphoglycerate, and no ATP can be formed. Figure 11.6 When we exercise slowly enough for blood circulation to keep up, all our muscular activity is aerobic, but with rapid, intense, and prolonged activity, blood does not carry oxygen to the muscles rapidly enough and lactic acid fermentation begins. Lactate accumulation causes cramps and muscle pain. Alcoholic fermentation involves the conversion of pyruvate to acetaldehyde before reduction by NADH plus H. Some goldfish survive low oxygen by respiring glucose to ethanol, for example, when ice on a lake surface prevents oxygen from dissolving into the water. Plants and people. Box 11-1 Fungal respiration, the prehistoric industrial revolution. Most college students know at least a little about fermentation. You probably already know that beer and wine are fermented even though you never much worried about the Embden-Meyerhof pathway. Perhaps you know already that aerobic respiration of the fungi known as yeast causes bread to rise in holes to form in Swiss cheese, but you may not have realized that the use of these fermentations started out as life-saving processes several thousand years ago. When we think about the origins of agriculture between 2000 and 300 BCE, we often think of the domestication of wheat, rice, and corn, but the use of fruits and animal products was necessary not only for diversity in the diet but also for diversity of vitamins and essential nutrients. Think about life in 300 BCE, no supermarkets, of course, and no canned food, frozen food, or refrigerated food. People grew their own food, or collected it, or traded for it. Now imagine the first cold winds and rains of autumn. How do you preserve the abundant food from summer to allow your survival through winter? It will be 7 long months before grains can be planted, before the dormant buds on grapevines open and put out their new leaves and flowers. Even after spring, the first crops cannot be harvested until May or June at the earliest. Hopefully, cows will give birth and there will be milk. How do you preserve food for the winter? Grains such as wheat dry naturally just before harvest and can be stored rather easily. They are so dry that microbes cannot grow. One problem is that they must be kept dry, protected not only from winter's dampness but also from the water produced by their own slow respiration. Fruits also can be dried, but these cannot be kept dry reliably without cellophane or plastic bags. Under ancient storage methods, dried fruit often drew enough moisture from wet winter air to allow fungi and bacteria to grow on them. One way to prevent microbial growth was discovered early: fermentation. If sugary fruits were partially respired in a sealed jar, production of ethanol would finally kill all microbes, sterilizing the fruit. As long as the jar remained sealed, the fruit was safe, including all of its minerals, vitamins, amino acids, and so on. Beer is most often fermented barley, and barley grows best in cool, damp, northern climates where it is most difficult to keep grains dry. Natural, unpasteurized milk becomes sour in just 2 or 3 days at room temperature as bacteria grow rapidly, but if the right fungi are added, milk is only partially degraded and the resulting cheese is dry and stable enough to stop bacterial growth. Covered with a layer of wax to protect it from airborne bacteria, it can last for years. Using yeast to leaven bread has basically the opposite effect. If wheat is ground to flour, mixed with water, and then baked, the resulting bread can be so hard and dry that it will last forever, even in your mouth. Mastication is an ordeal. However, carbon dioxide from aerobic respiration of baker's yeast becomes trapped by the bread dough, causing the bread's texture to be lighter and more open. This bread does not preserve as well, but it is easier to eat. Ethanol and lactate are not especially good solutions to the problem of NADH accumulation. The pyruvate consumed is always present in adequate amounts, of course, because glycolysis itself produces it, but it is not really cheap because many of its bonding orbitals have high-energy electrons. Furthermore, pyruvate could be used as a monomer for many types of synthesis. A lack of oxygen forces a cell to use a very valuable molecule as an electron dumping ground. Even worse, the products, either lactate or ethanol, are toxic. If they accumulate in the tissues or environment, they damage or even kill the cells producing them. Table 11, 2 electron acceptors. Figure 11, 7 in winemaking, yeast cells ferment the glucose present in grapes and excrete the waste product ethanol. Naturally fermented wine has a maximum alcohol content of 14%. At that point, the waste product kills the yeast. Beer has a lower alcohol content, 5%, not because its yeast is more sensitive to the ethanol but because it is the legal limit. Fermentation must be stopped artificially, usually by heating the beer to kill the yeast. Alcoholic beverages with alcohol contents higher than 14% have extra alcohol added or part of their water removed. The leaves and upright stems of these marsh plants exist in a highly oxygenated aerobic environment, but the rhizomes and roots exist in an anaerobic muck. Parenchyma tissues in the stems permit the diffusion of some oxygen from leaves to rhizomes and roots, but some anaerobic respiration may be occurring. The trees in the background only survive where there is adequate oxygen in the soil. Figure 11, 8 Even under flooded, anaerobic conditions, rice seeds germinate and begin to grow because their embryos carry out facultative anaerobic respiration. Other seeds remain dormant because they are incapable of fermentation. Being dormant, they have a low rate of metabolism and the small amount of oxygen present may keep them from dying immediately. When flooding subsides, oxygen is available and non-rice seeds germinate. However, they are shaded by rice plants and the soil is already filled with rice roots. The rice plants have a tremendous advantage. Rice seedlings, still flooded, Considering the negative aspects of anaerobic respiration, how could natural selection have produced something so inefficient? At the time life arose and respiratory pathways were evolving, Earth's atmosphere contained reduced, hydrogen-rich compounds but no oxygen. Consequently, pyruvate and acetaldehyde had to be used as electron acceptors. Millions of years later, photosynthesis based on chlorophyll evolved, and oxygen was released to the environment as a result of the water splitting involved. After free oxygen became relatively abundant, the mutations leading to aerobic respiration began to be selectively advantageous. Glycolysis must be one of the most ancient of all metabolisms. It occurs in all organisms, with no exceptions. Even in the presence of an oxygen-rich atmosphere and aerobic respiration, anaerobic respiration still has some selective advantage. Although the method is far from ideal, it does allow certain organisms to survive in particular environments. For example, because rice seedlings are capable of anaerobic respiration, they germinate and establish themselves during times of floods when other plants or seedlings are suffocating. In the absence of oxygen, the alternative to anaerobic respiration is death. Although this metabolism is expensive for the rice, by the time the flooding subsides, rice plants are already well-rooted, have several leaves, and outcompete other species whose seeds are just beginning to germinate. Alternatives. Box 11-2 Respiration in prokaryotes. Basic reactions. In all plants, animals, fungi, cyanobacteria, and most bacteria and archaea, the compounds used for respiration are organic, sugars, fats, amino acids, and so on. However, certain prokaryotes metabolize inorganic compounds and extract energy from them, examples being hydrogen, sulfur, iron, and nitrogen. Electron donors. Hydrogen respiration is easy to understand. The reaction 2H plus O2HO liberates 2 electrons and releases so much energy that it easily forces electrons onto NAD, producing NADH. This then donates the electrons to an ordinary electron transport chain that produces ATP. Hydrogen sulfide and thiosulfate can both be converted to sulfur and then to sulfate. These reactions release energy and electrons that either go into an electron transport chain or react directly to produce ADP. The end product is sulfuric acid, which is toxic like the ethanol produced in plant and yeast fermentation. As long as the environment absorbs the acid and keeps it dilute, the bacteria are unharmed. Thiobacillus thiooxidans grows well even at pH 2. Oxidation of ferrous iron to ferric iron releases an electron that can be used to form a single molecule of ATP. Likewise, ammonium can be oxidized to nitrite and then to nitrate, releasing 6 energetic electrons that can be used for ATP synthesis. Electron acceptors. Just as various prokaryotes use various reduced compounds as sources of electrons and energy, other prokaryotes use various highly oxidized compounds as electron acceptors. The most common is nitrate, which is converted to either NO or N. Sulfate also acts as an electron acceptor and is converted to hydrogen sulfide. These processes are the opposite of those described previously here, in which these same compounds were used as substrates for respiration because they could be oxidized. How can they act as a source of electrons one time but as acceptors another time? The key is the presence or absence of oxygen. Oxygen has such a strong tendency to absorb electrons that in its presence the balance between HS and SO is shifted in favor of SO. Without oxygen, the balance is shifted the other way. Certain organic compounds act as electron acceptors when oxygen is not present. The process is fermentation. Bacteria ferment diverse substrates such as numerous sugars and organic acids, resulting in products like ethanol, lactate, propionate, acetate, acetone, methanol, and carbon dioxide. Fermentations of amino acids and other compounds that contain nitrogen or sulfur are often called putrefactions and produce odoriferous compounds, hydrogen sulfide, isobutyric acid, cadaverine, putrescine, and ammonia. Researchers are experimenting with methods to increase fermentation of cellulose and lignin to ethanol or methane to be used as fuel, but as you know, these are extremely inert, non-reactive compounds. Figure B11, 2a. This stream in Colorado is in a natural condition with clean water. Figure B11, 2b. This stream is located only a few miles from the other, but it is polluted with runoff from mining wastes. Iron oxidizing bacteria use the ferrous iron as a substrate, respiring it to ferric iron and generating ATP. Virtually no other life occurs in such polluted conditions. Integrated metabolism. Relationships between photosynthesis, respiration, and carbon are simple but important. All photosynthetic plants are autotrophs. They take in carbon dioxide, reduce it to carbohydrate by photosynthesis, transport it to another part of the plant, and then use most to build structures. They respire the rest to produce ATP. All animals are heterotrophs. Their food consists of reduced carbon compounds, and a small amount is used to construct animal tissues. However, the majority is respired as an energy source. All animals lack the Calvin cycle. No animal is able to fix carbon dioxide. In prokaryotes, similar relationships hold and others also occur. Green bacteria, cyanobacteria, and some purple bacteria are photosynthetic and have the Calvin cycle. They are therefore photoautotrophs like plants. Most other bacteria, like animals, take in organic substances and use them both for ATP generation and for polymer construction. These are conventional heterotrophs. Photosynthetic bacteria, however, commonly have food and light available, and most purple bacteria and green bacteria are photoheterotrophs. They absorb and use organic carbon for construction and use photosynthesis almost exclusively to generate ATP. Little or no photosynthetic energy is used to reduce NADP to NADPH because NADPH is not in great demand, because the Calvin cycle is unnecessary if carbohydrate is present in food. Lithotrophs use carbon dioxide as their primary carbon source. They are lithotrophic autotrophs. Examples are the colorless sulfur bacteria, hydrogen bacteria, nitrifying bacteria, and iron bacteria. As they oxidize hydrogen, sulfur, iron, and so forth, the energy goes to make ATP, part of which is used for regular metabolism and part to pump protons, creating a pH gradient that can be used to reduce NADP to NADPH. Their energy metabolism is somewhat like bacterial photosynthesis. Neither metabolism can produce a compound as energetic as activated chlorophyll. A very few bacteria are lithotrophic heterotrophs. They get their energy by oxidizing sulfur compounds and they take in organic compounds for structural uses. They do not use carbon dioxide, and apparently they lack the Calvin cycle. The ability to live as a heterotroph offers great advantages. By taking in carbon in its reduced form, heterotrophs use all their respiratory or fermentative energy for growth and reproduction. Autotrophs, which must use energy to reduce carbon dioxide, seem to be at a disadvantage. Their one advantage is that an autotroph has probably never starved to death. Carbon dioxide is always abundant enough to sustain the life of autotrophs. The same is not true for the food supply of heterotrophs. Whether that food is organic or not, famine is a common occurrence not only for animals and fungi but also for bacteria. Aerobic respiration. With oxygen present, the problems of using pyruvate or acetaldehyde are eliminated. Oxygen is absorbed and acts as the terminal electron acceptor. Oxygen is inexpensive because it is absorbed and distributed by molecular diffusion, which requires neither active transport nor ATP consumption. Oxygen is abundant in most situations, and the product of reduction is water, which is not only non-toxic but actually beneficial. Aerobic respiration in plants is almost identical to that in animals, but we must be careful when comparing plants to ourselves. Our cells consist almost entirely of protoplasm, whereas plant cells consist mostly of a large central vacuole. A kilogram of muscle, for example, contains much more metabolically active protoplasm than does a kilogram of parenchyma. Consequently, a kilogram of plant tissue needs much less oxygen than does a kilogram of animal tissue. Earth's atmosphere now is 21% oxygen, but most plants survive with no trouble even if we artificially lowered the oxygen to 5%. Many plants thrive even in air with only 2% oxygen. Under ordinary circumstances, herbs, shrubs, and trees growing in regular air have about 250 times more oxygen than they need for full aerobic respiration. Aerobic respiration consists of three parts: glycolysis, the citric acid cycle, and oxidative phosphorylation in an electron transport chain. Glycolysis. The initial steps of aerobic and anaerobic respiration are identical: glycolysis by the Embden-Meyerhof pathway to pyruvate. This produces ADP and NADH just as before, but with oxygen present. The NADH migrates to electron carriers that oxidize it back to NAD, permitting glycolysis to continue. Glycolysis occurs in the cytosol and plastids. The citric acid cycle. Because pyruvate is not needed as an electron acceptor, it can be used in a number of metabolic pathways. One of the main pathways takes advantage of the large amount of energy remaining in pyruvate and rather than using it for its structure, breaks it down and generates more ATP. This one pathway has three names: the citric acid cycle, the Krebs cycle, or the tricarboxylic acid cycle. These names reflect different facts about the cycle. One of the intermediates is citrate, the anion of citric acid. Much of the pioneering work on this metabolism was carried out by Hans Krebs. Finally, several of the intermediates are tricarboxylic acids, that is, each has three carboxyl groups. Figure 11.9 The process by which pyruvate is attached to CoA releases a carbon dioxide molecule and forms NADH. This step does not occur during anaerobic respiration. Not only would it use up pyruvate needed as an electron acceptor, but it would generate even more NADH. Both of those results would be detrimental under anaerobic conditions, but both are beneficial when oxygen is present. In the citric acid cycle, pyruvate is transported from the cytosol, where glycolysis occurs, across the mitochondrial membranes to the mitochondrial matrix. There it is oxidized and decarboxylated, as electrons are transferred to NAD, bonding orbitals holding the last COO rearrange and CO is liberated. Carbon dioxide and NADH are produced, along with a 2-carbon fragment called acetyl. The carbon dioxide and NADH remain free in the matrix solution, but the acetyl becomes attached to a carrier molecule, coenzyme A. The resulting combination being acetyl-CoA. Like pyruvate, acetyl-CoA can be used in many synthetic pathways, but here we are interested in its entry into the citric acid cycle by transfer of the acetyl group to an acceptor molecule, oxaloacetate, a compound with 4 carbons. The oxaloacetate is converted to a 6-carbon compound, citrate, which is then rearranged to cis-aconitate, which in turn is transformed to isocitrate. In the next step, one of the carbons of isocitrate is oxidized by passing electrons onto NAD, creating NADH. The oxidized carbon is liberated as carbon dioxide, leaving alpha-ketoglycerate, which has only 5 carbons. This too is oxidized by NAD, liberating another carbon dioxide, and the 4-carbon remnant becomes attached to a new molecule of CoA in the process, forming succinyl-CoA. The energy released by the breakdown into free CoA and free succinate can power phosphorylation of ADP to ATP. The succinate still contains considerable energy and is oxidized to fumarate as electrons and protons are passed to flavin adenine dinucleotide, reducing it to FADH. Although the molecule becomes oxidized by this step, no carbon dioxide is lost. The fumarate is also a 4-carbon compound. It reacts with a water molecule and becomes malate, which passes a final set of electrons onto NAD and is transformed into the original acceptor molecule, oxaloacetate. It was mentioned above that the benefit of the citric acid cycle is the generation of more ATP. Nevertheless, at only one step has ATP been produced. Instead, there are 4 steps in which more NAD is reduced to NADH and 1 in which FAD is reduced to FADH. Excess NADH and the related deficiency of NAD were seen to be problems in anaerobic respiration, and thus, the citric acid cycle at first seems to be a real contradiction. However, in the next step, the electron transport chain, the energy in NADH and FADH drives the synthesis of ATP, and NADH is simultaneously oxidized back to NAD. The mitochondrial electron transport chain, chemiosmotic phosphorylation. The mitochondrial inner membrane, like the chloroplast inner membrane, contains sets of compounds capable of carrying electrons. Although many of the actual carriers differ in the two organelles, the principles of electron transport are the same. The carriers that react with NADH or oxygen are placed precisely and asymmetrically in the crystal membranes, both on the matrix side only. The exact order of carriers in plant mitochondria is complex, and numerous types exist. Only the most well understood are presented later here. NADH diffuses to the membrane and passes electrons to a protein that has flavin mononucleotide bound to it as a cofactor. The FMN is reduced, and the NADH simultaneously oxidized to NAD, which can migrate back to the site of the citric acid cycle. Reduced FMN passes the electrons to a set of iron and sulfur-containing, proteinaceous electron carriers, which transfer the electrons to one or several quinones, one of which is ubiquinone. From the pool of quinones, electrons are transferred to cytochrome B. The next carriers in sequence are more quinones, then cytochrome C, cytochrome A, and cytochrome A. Cytochromes A and R are part of a large enzyme complex known as cytochrome oxidase, which contains several proteins and 2 copper ions that mediate transfer of electrons from the iron in cytochrome to oxygen. As oxygen is reduced, it picks up 2 protons and becomes water. If this electron transfer were the only thing to happen in the electron transport chain, it would be better than anaerobic respiration because the cell could avoid the waste of pyruvate and the synthesis of the toxic lactate or ethanol, but much more is accomplished. Mitochondria also perform chemiosmotic phosphorylation. As with chloroplasts, the fate of the protons released and absorbed during electron transport is important. In mitochondria, when NADH reacts with FMN, 2 protons are transferred as well as electrons. 1 proton comes from the NADH and the other from the water, both on the matrix side of the membrane. When FMNH reduces any quinone, the 2 protons are released on the other side of the membrane into the lumen of the crista. This step of electron transport acts as a proton pump. A large concentration of protons begins to build up in the lumen while there is a lack of protons in the matrix. Ubiquinone and the other quinones just before cytochrome C act in a similar manner, pumping protons out every time they carry electrons. Also, as oxygen receives electrons from the electron transport chain, it forms water by picking up 2 protons from the matrix, decreasing the proton concentration. In a short time, a proton concentration gradient develops that is strong enough to cause the protons to migrate back into the matrix. Figure 11, 10 The steps of the citric acid cycle. This cycle is an important part of aerobic respiration, even though it does not consume oxygen and generates only 1 molecule of ATP directly. DH equals dehydrogenation, DC equals decarboxylation. Figure 11, 11-flavin adenine dinucleotide is a large organic electron carrier similar to NAD. When it is reduced by 2 electrons, it picks up 2 protons, becoming FADH. Unlike NADH plus H, both protons are covalently attached to FADH. The flow of protons from the crystal lumen to the mitochondrial matrix can be used to synthesize ATP. As in chloroplasts, ATP synthetase channels in the membrane use the flow of protons to force a phosphate group onto ADP, creating ATP. This is a chemiosmotic phosphorylation, and because of it, the NADH is an excellent if indirect source of ATP rather than a problem. The electrons that each molecule of NADH contributes to the mitochondrial electron transport chain provide enough power to create 3 ADPs. FADH also passes its electrons to the mitochondrial electron transport chain, but it reacts with ubiquinone instead of FMN, thus, the first step in proton pumping does not use these electrons. Therefore, FADH contributes less to the proton gradient than does NADH, providing enough power for production of 2 ADPs rather than 3. Figure 11, 12 membrane-bound electron carriers of the mitochondrial electron transport chain. As in chloroplasts, their positions and movements are important. Electrons are brought to membrane-bound carriers by mobile carriers such as NADH and FADH, which are produced in the matrix. Figure 11, 13. Only the important components of the mitochondrial electron transport chain are shown in this diagram, the steps that transport protons from the matrix to the crystal lumen, establishing a proton hydroxyl chemiosmotic gradient, just as in chloroplasts. The formation of water contributes to the gradient because protons are absorbed from the matrix but not from the cristae. FMN is flavin mononucleotide, an electron carrier similar in structure to FAD. The NADH shuttle. NADH produced by glycolysis cannot cross the mitochondrial inner membrane and donate electrons directly to the electron transport chain because the inner membrane is impermeable to such large molecules. Instead, a series of chemical reactions carries, reducing power across the membrane. Several shuttle mechanisms occur. In the malate aspartate shuttle, NADH in the cytosol powers the conversion of oxaloacetate to malate, which crosses to the mitochondrial matrix and powers the formation of a new molecule of NADH. Malate is converted to aspartate and transported back out of the mitochondrion, where it is converted to oxaloacetate again and can repeat the cycle. In this shuttle, each cytosolic NADH drives the formation of a matrix NADH and the consequent oxidative phosphorylation of 3 ADPs to 3 ADPs. In a second type of shuttle, the glycerol phosphate shuttle, cytosolic NADH reduces dihydroxyacetone phosphate to glycerol phosphate, which is transported across the inner membrane to the matrix. There it converts back to dihydroxyacetone phosphate, reducing FAD to FADH in the process. Each cytosolic NADH results in the formation of one matrix FADH, which can drive the formation of only 2 ATPs. Not as much energy is conserved in this shuttle. Figure 11, 14. Ubiquinone is a quinone that can carry 2 electrons and 2 protons simultaneously. Plants do things differently. Box 11 to 3. Plants, babies, and heat. Most plants resemble cold-blooded animals in having a temperature close to that of the immediate environment and lacking thermogenic mechanisms. Plants are not known to have any ability to sense their own temperature, and the few plant tissues and organs that do heat up do so in response to their own developmental state, such as being ready to open their flowers. In contrast, warm-blooded animals like us humans monitor our internal body temperature, core temperature as opposed to skin temperature, by the hypothalamus portion of our brain. It can detect changes in core temperature of as little as 0.01 degrees Celsius, and when core temperature drops, the hypothalamus causes heat generation. A strange thing is that we warm-blooded animals do not have a direct method of thermogenesis. All we can do is shiver, rapidly contracting and relaxing our muscles and wasting ATP just for the heat released. The few plants that generate heat do so more directly, using thermogenic respiration, which transports electrons in mitochondria without pumping protons and without saving any of the energy as ATP. Newborn human babies have a thermogenic mechanism rather similar to that of plants. They have a special type of fat, called brown fat, in which the cells are filled with numerous mitochondria, but these mitochondrial membranes are permeable to protons. Mitochondrial electron transport occurs, along with proton pumping. However, no chemiosmotic gradient builds up, and no ATP is synthesized. Thus, all energy is converted to heat. This is an extremely efficient method of generating heat, but for some reason, we give this up when we are several months old. While still babies, we acquire the ability to shiver and lose all of our brown fat. Mammals that hibernate, however, retain brown fat thermogenesis. Hibernating bears stay warm just like human babies, and almost like plants. Brown fat cell and plant thermogenic respiration have similar results, the generation of heat. It is not known whether one is more suited to animals and the other to plants. Perhaps in animals the mutation of proton-permeable membranes occurred earlier than mutations of the genes for alternative carriers, whereas in plants the opposite occurred. In at least some plants, NADH can cross the outer mitochondrial membrane and react directly with ubiquinone at the outer surface of the inner membrane. A shuttle mechanism is not necessary, but the step of proton pumping by FMN is bypassed, decreasing the amount of ATP that can be generated. Figure 11:15. The malate, aspartate shuttle allows reducing power to be transferred into mitochondria when NADH itself cannot cross the mitochondrial membranes. Heat-generating respiration. During glycolysis, the citric acid cycle, and mitochondrial electron transport, small amounts of energy are lost in each step even though a great deal of energy is conserved by the synthesis of ATP. The total chemical energy of all products is less than that of all reactants. The difference is lost as heat and increased entropy. For example, compost piles become warm because of the heat loss during the respiration of the fungi and bacteria that decompose the compost. Heat loss is usually an inefficient aspect of respiration. In most cases, it would be selectively advantageous for a plant to produce more ATP from each molecule of glucose respired and lose less heat to the environment. The heat loss during respiration by warm-blooded mammals is vitally necessary to maintain body temperature, and when we are chilled, we must generate even greater amounts of heat by shivering. Our muscles contract and relax rapidly, breaking down large amounts of ATP and releasing the stored energy as heat. Some plants also generate large amounts of heat. In the voodoo lily, parts of the inflorescence become much warmer than the surrounding air, causing amines and other chemicals to vaporize and diffuse away as chemical attractants for pollinators. Skunk cabbage often begins floral development while covered with snow. It melts the snow cover and exposes its flowers by generating large quantities of heat. These and other plants produce heat much more efficiently than humans do. They have alternative electron carriers that apparently do not pump protons during electron transport in mitochondria. Consequently, there is no proton gradient and no chemiosmotic production of ATP. The energy in NADH is converted entirely to heat, and the tissues become quite warm. Figure 11:16. By generating heat internally, skunk cabbage maintains a high enough temperature to carry out active metabolism even when covered by snow. When it is ready to emerge, it produces even more heat and melts the snow, revealing the inflorescence to pollinators. In ordinary mitochondria, cyanide, azide, and carbon monoxide interfere with the last electron carrier, cytochrome oxidase. When they are present, electrons cannot pass from cytochrome C to oxygen. Without electron flow and consequent ATP generation, both animals and plants die unless capable of anaerobic respiration. In plants that generate heat, the alternative electron carriers do not interact with cyanide, azide, or carbon monoxide, and heat is generated even if these chemicals are present. Heat-generating respiration can thus be studied by poisoning normal aerobic respiration with cyanide. Consequently, heat-generating respiration is usually called cyanide-resistant respiration. A better name is thermogenic respiration. The term cyanide-resistant respiration is somewhat misleading because it suggests that plants can be immune to cyanide poisoning. The analogy that because anaerobic respiration allows cells to survive in anaerobic conditions, cyanide-resistant respiration must allow cells to survive in the presence of cyanide is incorrect. Plants never encounter high concentrations of cyanide in natural conditions. If they did, they would be killed because aerobic ATP generation is blocked. Thermogenic respiration and heat-generating respiration are less confusing terms. Many aspects of thermogenic respiration are still unknown. The alternative carriers appear to be present in all mitochondria, and they seem to be resistant to stressful conditions such as cold or desiccation. One hypothesis is that during stress, the ordinary, proton-pumping carriers do not function as well, causing ATP production to drop, but the alternative carriers continue to transport electrons, allowing NADH to be converted back to NAD, which can then be used to keep glycolysis running and producing ATP. If this hypothesis is correct, thermogenic respiration is a metabolism that typically functions only rarely and helps plants survive stress conditions, and it has been adapted for actually generating heat in a small number of species. Pentose phosphate pathway. The intermediates of all respiratory pathways can be used in other pathways to make various compounds. It is not inevitable that they will be completely oxidized to carbon dioxide and water. The pentose phosphate pathway, which involves several intermediates that are phosphorylated 5-carbon sugars, is an important source for many fundamental compounds. It is usually included in discussions of respiration because it begins with glucose 6-phosphate, gives off carbon dioxide, and involves oxidations that produce NADPH. However, its importance as a source of respiratory energy is much less significant than its role as a synthetic pathway. The pentose phosphate pathway transforms glucose into 4-carbon sugars and 5-carbon sugars that are essential monomers in many metabolic pathways. The ribose 5-phosphate produced can be shunted into nucleic acid metabolism, forming the basis of RNA and DNA monomers, the nucleotides. In meristematic cells, large amounts of DNA must be synthesized during the S phase of a short cell cycle. The pentose phosphate pathway is an extremely important part of the metabolism of these cells. Figure 11:17. If NADH breaks down to NAD, a large amount of energy is liberated. If this powerful exergonic reaction is not coupled to any endergonic reaction, all of the energy is converted to heat. When coupled to the endergonic synthesis of ATP from ADP, most of the energy is conserved and little is converted to heat. The 4-carbon sugar erythrose 4-phosphate is the starting material in the synthesis of many compounds. Two important types are lignin and anthocyanin pigments. Tissues such as wood, fibers, and scleriates deposit large amounts of lignin into their secondary walls during development, and erythrose 4-phosphate is in great demand. During differentiation, these cells use the pentose phosphate pathway but pull out erythrose 4-phosphate, not ribose 5-phosphate. Flower petals and brightly colored fruits divert erythrose 4-phosphate from the pentose phosphate pathway to anthocyanin production while synthesizing their pigments. The pentose phosphate pathway also occurs in plastids, where it supplies erythrose 4-phosphate for synthesis of amino acids such as tyrosine, phenylalanine, and tryptophan. In addition to these 4 and 5-carbon sugars, the pentose phosphate pathway also produces NADPH. Although many anabolic reductions in the cytoplasm use NADH rather than NADPH, the reduction of nitrate to amino acids can be accomplished only by NADPH, which can be generated by the pentose phosphate pathway. Many of the reactants in the pentose phosphate pathway are the same as those in glycolysis, and both pathways occur in the cytosol and plastids. They are best understood as interconnected and simultaneous pathways. In meristematic cells, the pentose phosphate pathway is active and shifted in favor of ribose production. In wood cells, it is also active but produces erythrose. In other cells, it may be much less active and glycolysis may dominate, producing NADH that then powers ADP production in mitochondria. Imagine a cell as it is produced in the vascular cambium and then differentiates into lignified wood parenchyma, which after the wall is mature, takes on the role of storing starch during the summer and releasing it in the spring. Energy metabolism is adjusted at each stage in a major way, and smaller changes in the rates of reactions may occur on a day-to-day basis. Figure 11:18. The pentose phosphate pathway, showing all intermediates. If ribose 5-phosphate is drawn off into nucleic acid metabolism, the pentose phosphate pathway is shifted in favor of ribose production. If erythrose 4-phosphate is diverted to lignin metabolism, the pentose phosphate pathway reaction equilibria are shifted toward erythrose production. Figure 11:19 at the cambium, the pentose phosphate pathway may be producing ribose 5-phosphate, but in the differentiating vessels, it is producing erythrose 4-phosphate. In all cells, glycolysis and the rest of aerobic respiration are occurring simultaneously with the pentose phosphate pathway. In petals, erythrose 4-phosphate, used in the production of pigments, is produced by the pentose phosphate pathway. Respiration of lipids. Some tissues, especially oily seeds and dormant apical meristems, store large amounts of lipid, usually as triglycerides or phospholipids. During germination or release from dormancy, lipids undergo catabolic metabolism in which they are broken down into glycerol and 3 fatty acids or glycerol phosphate and 2 fatty acids. Fatty acids are then further broken down into 2 carbon units, acetyl-CoA, by a process called beta oxidation in either cytosol or microbodies called glyoxysomes. For example, an 18-carbon fatty acid would be converted into 9 acetyl-CoA units. As each acetyl-CoA is formed, one FAD is reduced to FADH, and one NAD is reduced to NADH, both of which can carry electrons to mitochondria and drive production of ADP by means of the electron transport chain. Acetyl-CoA may be used for synthesis of carbohydrates and other compounds, or it may enter the citric acid cycle and be further respired. Figure 11:20 All 20 amino acids are constructed from intermediates of respiration. Although too much sugar harms our health, plants construct everything from glucose and some minerals. Photorespiration. Photorespiration occurs only when Ruby-P carboxylase adds oxygen rather than carbon dioxide to ribulose 1,5-bisphosphate, resulting in one molecule of 3-phosphoglycerate and one of phosphoglycolate. Phosphoglycolate is dephosphorylated to glycolate, which is then transported to microbodies called peroxisomes. Glycolate can be converted to glycine in the peroxisomes, and the glycine may be transferred to mitochondria, where it is respired. To carbon dioxide and water with no conservation of energy in either ATP or NADH, all energy is wasted. In some cases, some of the glycine can be converted to serine. Both are useful amino acids. Other mechanisms also produce these amino acids, however, and direct measurements show that for many sea species, photorespiration wastes as much as 30% of the energy trapped by photosynthesis. Environmental and internal factors. As with photosynthesis, numerous environmental factors influence the rate of respiration. It is necessary to consider the integration of respiration into the total biology of the plant as well as the metabolic pathways involved. Temperature. Temperature greatly influences respiration in a plant growing under natural conditions. In all environments, shoots located in air are at a different temperature than roots in soil. Shoots are subjected to great changes of temperature from day to night, often more than 20 degrees Celsius. In most tissues, an increase in temperature of 10 degrees Celsius, in the range between 5 degrees Celsius and 25 degrees Celsius, doubles the respiration rate, a magnitude of increase seen in many enzyme-mediated reactions. Below 5 degrees Celsius, respiration decreases greatly. Above 30 degrees Celsius, respiration still increases, but not so rapidly. At such high temperatures, oxygen probably cannot diffuse into tissues as rapidly as the tissues use it. Above 40 degrees Celsius, respiration, like many other processes, slows greatly, probably because of enzyme damage or disruption of organelle membranes. Lack of oxygen. Because plants are not as active as animals, much lower oxygen concentrations, as little as 1% to 2%, maintain full rates of plant respiration. Oxygen concentration in the atmosphere is so stable that it does not cause variations in respiration, but variations occur in a cell's access to oxygen. During daylight hours, chlorophyllous tissues and organs produce oxygen, some of which is used in respiration. At night, oxygen is not produced, but it can diffuse into the large intercellular spaces of the plant if it can penetrate the closed stomata. Because the cuticle is not absolutely impermeable to oxygen and because the atmosphere contains such a high concentration, oxygen diffuses in rather well, even through closed stomatal pores. Less oxygen is available, but the temperature is lower, and thus, demand for oxygen is reduced. Oxygen availability is much more variable for roots. In well-drained soil, the large quantity of gas located between soil particles is depleted in oxygen owing to respiration by roots, fungi, bacteria, protists, and soil animals. During and after rain, soil air is displaced by water, and roots either have lesser amounts of oxygen or none available. Anaerobic respiration allows roots to survive for a short period, but it is not sufficient for root growth and healthy metabolism. If continually flooded, roots of almost any species die. Those that survive do so primarily by oxygen diffusion from the stems through parenchyma channels in the cortex or pith, such as in petioles of water lilies and cattails. Floods that submerge the bases of shoots or woody trunks are especially damaging if they last more than a few days. Rice seedlings are the only certain example of plants that can grow normally without oxygen for a prolonged period. Figure 11:21 During the respiration of lipids, fatty acids are separated from glycerol and undergo beta oxidation to acetyl-CoA units, producing FADH and NADH. In germinating seeds, much of the acetyl-CoA is converted to glucose and fructose by gluconeogenesis, then are polymerized into sucrose. The sucrose is transported by phloem from cotyledons to embryo meristems, where it is used in construction or respired for energy. Figure 11:22 Trees in cold winter weather are not necessarily dormant. Sunlight warms them enough for metabolism to occur. Although respiration rates are low, it provides enough energy for critical metabolic reactions. The conifers still have chlorophyll present, so even photosynthesis may be possible. In thick tubers or bulky roots, it is not well established whether respiration is completely aerobic or at least partially anaerobic. Such organs usually have a significant amount of intercellular space through which oxygen might diffuse, but the concentration of oxygen is very low. Similarly, sapwood and cambium of trees with thick bark may be hypoxic. Even though lenticels are present, the diffusion path is long, and the dense inner bark, cambium, and sapwood lack intercellular spaces. Developing embryos inside large seeds and fruits are reported to respire anaerobically. Internal regulation. Like virtually all other processes, respiration is subject to specific metabolic controls. Cells that have an active metabolism, such as glands that secrete protein or epidermal cells that secrete waxes, have a high level of aerobic respiration, whereas meristematic cells produce ribose by means of the pentose phosphate pathway. Nearby cells, perhaps those of the spongy mesophyll or parenchyma, may have a much lower respiration rate. During fruit maturation, respiration usually remains steady or increases gradually until just before the fruit is mature, at which point a sudden burst of respiration is triggered by endogenous hormones. Conversely, within seeds, after an embryo is mature, its respiration decreases so dramatically that it is difficult to measure, and the seed becomes dormant. In seeds with a true dormant period, virtually no respiration occurs even if the embryo is surgically removed and given water, warmth, and oxygen because metabolic inhibitors suppress respiration. Although chloroplasts produce large amounts of ATP while photosynthesizing, apparently none of that is exported to the cytoplasm. Respiration is the source of ATP for all parts of a cell other than chloroplasts. Total energy yield of respiration. During anaerobic glycolysis, 4 molecules of ATP are synthesized, whereas either 1 or 2 ATPs must be used to initiate the process, depending on whether glucose or glucose 6-phosphate is the initial substrate. The NADH plus H generated cannot be used for energy, and thus, the net result is 2 molecules of ATP for every molecule of glucose fermented. Table 11:3. During aerobic respiration, glycolysis again yields 2 ATPs directly. In addition, the 2 NADs can be transported to mitochondria, where their electrons power the formation of 2 or 3 more ATPs. The conversion of each pyruvate to acetyl-CoA yields another NADH. Because 2 pyruvates are produced from each initial glucose, 6 more ATP per glucose are produced. Within the citric acid cycle, each original molecule of glucose yields 2 molecules of ATP, 6 of NADH, and 2 of FADH. The total is 24 ATPs. Aerobic respiration can produce as many as 38 molecules of ATP, making it significantly more efficient than anaerobic respiration. Figure 11, 23 respiration rates for various parts of loblolly pine trees in a 14-year-old plantation. Respiration is much higher in summer than in winter for all organs, and leaves have the highest rate, 20 times the scale for the other organs. The rate is given as grams of carbon dioxide released per square meter of soil surface per hour. This type of measurement is more useful to a forest manager than to a cell physiologist. Different scientists or studies need different types of measurements of the same function. A cell physiologist might express respiration as grams of carbon dioxide released per cell per hour, per gram of protein per hour, or per gram of tissue per hour. Each gives a different result. Leaves have small cells with thin walls, so a gram of leaf tissue has much protein and many cells, whereas a gram of woody stem or branch is mostly inert, non-respiring cellulose walls with few cells and little proteinaceous protoplasm. It can be difficult to select an appropriate basis for stating rates of respiration, and comparing studies that use different bases is not easy. Table 11, 3 Production of ATP by various types of respiration. The pentose phosphate pathway yields only 2 NADPH per glucose 6-phosphate if either ribulose 5-phosphate or erythrose 4-phosphate is drawn off for anabolic metabolism. If neither of these is removed, the various intermediates continue to cycle until all carbon of the glucose is completely oxidized to carbon dioxide and 6 NADPHs are produced. These may be used for cellular reductions of nitrate to amino acids, sulfates to sulfhydryls, or carbohydrates to fats. NADPH not consumed in anabolic reductions may contribute protons to the mitochondrial electron transport chain and indirectly produce 2 molecules of ATP. The amount of ATP produced by fatty acid respiration depends on the length of the fatty acid and whether acetyl-CoA enters the citric acid cycle. With maximum respiration, up to 40% of the total energy in a fatty acid is conserved in ATP. Thermogenic respiration and photorespiration produce no ATP. Also, keep in mind that intermediates may be diverted from all respiratory pathways and be used in synthetic reactions, so complete respiration may not occur. ATP production thus is less. Table 11, for respiratory quotients of various compounds. Respiratory quotient. An action spectrum is a valuable tool for studying light-mediated phenomena such as photosynthesis. For respiration, a similar type of information is useful. A theoretical calculation can be made of the amount of oxygen consumed by each type of respiratory substrate. For example, the complete aerobic respiration of glucose should consume 6 molecules of oxygen and produce 6 molecules of carbon dioxide. This ratio of carbon dioxide liberated to oxygen consumed is known as the respiratory quotient. For glucose RQ equals 1. 0. Acids can enter the citric acid cycle and be oxidized to carbon dioxide, producing NADH for the mitochondrial electron transport chain. Because acids contain relatively large amounts of oxygen in their molecular structure, less is needed to convert them to carbon dioxide and water, so their RQ is high, greater than 1. 0. On the other hand, fatty acids contain virtually no oxygen, therefore, when they are respired, enough oxygen must be consumed to oxidize not only every carbon but every hydrogen as well. Thus, the RQ is very low, often about 0.7. Of course, anaerobic respiration consumes no oxygen, although its carbon dioxide production is very high. After these RQ values are calculated, it is relatively easy to measure the amounts of gases exchanged during respiration and thus gain information about the respiratory metabolism. For example, peanut seeds are rich in lipids and oils. As they germinate, the seedling's RQ is initially low, indicating that the lipids are being respired. If they are kept in the dark, the RQ remains low until the seedling exhausts its nutrient reserves and dies. But if they are allowed to germinate in the light, the RQ gradually increases toward 1.0 as photosynthesis begins to produce glucose needed for respiration. The increase is not immediate because much of the first products of photosynthesis are used for constructive anabolic reactions in the leaf cells, so a rise in the RQ indicates that leaf photosynthesis is meeting anabolic needs and producing extra for respiration. Figure 11, 24 sunflower seeds store oils in their cotyledons. When they germinate, the oils are respired, giving the seed a low RQ value. The cotyledons of the seed, although still enclosed by the fruit, have turned green and are carrying out photosynthesis, so both oils and sugars are available for respiration. After a few days, all the oil will be consumed, then only photosynthetically produced sugars will be available, and the seedling will have a high RQ value, near 1.0. Fermentation of alcoholic beverages. The ethanol of alcoholic beverages is always produced by the fermentation of glucose by yeasts. Sugars present in fruits can be fermented immediately, but starches in seeds and tubers must be depolymerized to glucose before they can be fermented. Ethanol produced by fermentation finally kills the yeast if it builds up to a concentration of about 18%. To obtain a stronger concentration of alcohol in the beverage, the fermented mixture must be distilled, a process in which it is heated to concentrate the alcohol while removing some of the water. Alternatively, extra alcohol can be added to a beverage rather than distilling it. These factors combine to give us three basic types of alcoholic beverages: beers, resulting from partially fermenting starchy seeds; wines, produced by a more complete fermentation of sugary fruits; and spirits, in which their ethanol concentration is increased by distillation or by adding alcohol. Beer. Beer is made by fermenting starchy cereal grains, especially barley, wheat, corn, or rice. Barley is by far the most common type of beer, and if a different grain is used, it is typically specified. For example, beers made from wheat are becoming more popular and are always called wheat beer. Before a cereal grain can be fermented, at least part of its starch must be converted to glucose. This is done by moistening the grains and allowing them to germinate. The moistened embryo secretes enzymes that break down the starch located in endosperm. Barley has the greatest number of enzymes and converts starch to sugar more quickly than do other grains once the seedling root is visible, sprouting has proceeded long enough, and the grains are dried to prevent any further enzymatic reactions. Sprouted, dried barley grains are called malt. To brew the beer, malt is mixed with water in a large vat. The mixture is called the mash. If malt is the main ingredient, the beer will have a strong flavor, and the quality of the sprouting and roasting of the barley is very important. But often, at least in the United States, unsprouted grains or even corn syrup or potatoes are added. These are very cheap sources of extra starch that can be acted upon by the enzymes in the malt. By adding these adjuncts, the beer is less expensive and typically has less flavor. Hops are the dried carpellate inflorescences of a tall, vining plant, Humulus lupulus, closely related to cannabis, marijuana. Hops have a bitter taste and aroma and are added so beer will not be too sweet. The greater the amount of hops, the more bitter the beer. Once all the ingredients are in the mash, it is warmed to 68 to 73 degrees Celsius and allowed to stand for several hours while the various enzymes are active. The chaff and solid materials are then strained out and the sugar-rich liquid, called wort, is boiled to inactivate the enzymes and kill any microbes. Once it has cooled, yeast is added. For beers that will be alesh, bitters, or stouts, Saccharomyces cerevisiae is used. but for lagers and pilsners, S. uvarum is added. Most beers in the United States are lagers. The yeasts are allowed to ferment the wort from 1 week to 12 days. The beer is filtered and pasteurized to stop all further fermentation and then is aged for 2 or 3 weeks. The carbon dioxide produced by fermentation is allowed to escape during brewing, so to make beer frothy when it is poured, new carbon dioxide is added to the beer artificially just before it is bottled. Various types of beer result from controlling the malting process, the adjuncts, hops, and the type of yeast used. S. cerevisiae, the yeast used for alesh, floats to the top of the fermenting vats, and it works best at a warm temperature. This species of yeast produces compounds called esters that add light, fruit-like flavors to the beer. Common types of ale are brown ale, pale ale, and porter. S. uvarum, used for lager beers, sinks to the bottom of the vats and functions at a cool temperature. Cool, bottom fermentation does not produce esters, so lager beers have a crisper taste. Most of the beer consumed in the United States is pale lager, with an alcohol content of only 3% to 6%. The concentration of alcohol depends on the amount of starch converted to fermentable sugar and whether fermentation is artificially stopped at a particular point. With a greater conversion of starch to sugar and a longer fermentation, the concentration of alcohol increases. Light beers have fewer calories than ordinary beer. This can be achieved in either of 2 ways. One is to begin brewing with a mash that contains fewer carbohydrates. When brewing is halted, regular beer still has many starches and unfermented sugars present, but light beer has fewer of them. This gives the beer fewer calories. Many people believe that most calories in beer come from the alcohol, which may be true for light beers but not for regular beers, and certainly not for stouts. These have relatively high amounts of starches and sugars, giving the beer its body and fullness. The second way to make light beer is to convert more of its starches to sugars. This ferments to a beer with much higher alcohol content but low in starch. Water is added at the end to lower the alcohol content to the normal 3% to 6% level for beer, but it also dilutes the flavor. Sake is fermented rice. It is usually called rice wine, but because it is a cereal, technically it should be classified as a beer. However, sake is made by a unique process that differs from brewing beer. The rice is polished to remove all fruit and seed coats and then is steamed to cook and soften its starch grains, as if it were to be eaten. Instead, it is inoculated with the fungus Aspergillus oryzae and allowed to set for several days. Enzymes from the Aspergillus depolymerize the starch to fermentable sugars. After the rice gets a distinctly moldy aroma, it is mixed with warm water, and then S. cerevisiae is added. No hops or adjuncts are added as they would be for beer, and instead of fermenting for only a week, sake is fermented for almost a month. During this time, the Aspergillus enzymes continue to convert more starch to sugar, and the yeast ferments that to ethanol. At the end, the sake is separated from the solids by filtration through cloth, and extra ethanol is added to bring the final concentration up to about 20% to 22%. This makes it a fortified beer. Sake is thus much stronger than beer, and it is served hot. Plants and people. Box 11 to 4 Respiration. For the most part, people do not affect plant respiration very much, and plant respiration does not have too much of an impact on people. Food storage is one aspect of respiration in which plants and people influence each other. Stored fruits and vegetables respire if they are alive, and if they are moist like fresh potatoes, carrots, peaches, and lettuce they respire especially rapidly. Even dry foods like beans, lentils, wheat, and rice respire, but very slowly. Frozen and canned foods are dead and do not respire. Respiration in dry foods is important because of the water produced. Remember that the formula for respiration is CH12O plus 6O6CO plus 6HO. This water is released from the mitochondria and moistens the cell's protoplasm, causing the food to become wet and thus susceptible to attack by fungi. Even if not attacked by fungi, the extra moisture may cause the cells to become more active metabolically, and seeds might even germinate while in storage. To keep wheat, corn, and similar foods dry, air is blown through the storage containers to remove any moisture produced by respiration. Figure B11-4a. In moist stored foods, the extra moisture produced by respiration is not much of a problem. A fresh watermelon has so much water already that the little bit produced by respiration is insignificant. Instead, the problem is that respiration uses up part of their carbohydrates, the very reason we cultivate and harvest most of them. Each week that these plants are stored, there is less carbohydrate available to us when we eat the food. A fresh potato is packed with starch, whereas a potato that has been stored for months has respired away much of its starch and is much less nutritious to us. Respiration in such foods is minimized by keeping them cool. Low temperatures slow down metabolism and thus slow down respiratory loss of carbohydrates. Sometimes we harm plant respiration indirectly, without even realizing it. We pollute rivers and lakes with fertilizers. This happens when we dump sewage, even cleaned up sewage, into rivers. The wastes we flush down our toilets are food to many types of bacteria, and such bacteria are able to grow rapidly and respire rapidly. With enough human fertilizer, the bacteria proliferate and use up most of the oxygen available in the water or in the mud on the bottom of a lake or river. Plant roots in those areas may then suffer from hypoxia or anoxia, and if the roots die, the plants may die also. Of course, fish need even more oxygen than roots do, so hypoxia caused by excessive bacterial growth may cause fish to die. A current problem is that the Mississippi River carries huge amounts of human waste, as well as that from farm animals such as cattle, pigs, and chickens. In addition, it also is polluted with agricultural fertilizers that are washed out of fields by rain. As the Mississippi's water reaches the Gulf of Mexico, it fertilizes the bacteria there. The bacteria grow and use so much oxygen that no fish or algae are able to survive. Our pollution causes a huge dead zone in the Gulf of Mexico. Similar dead zones occur in other oceans where rivers drain fertilizer-enriched water from farms and cities. Figure B11, 4a grains such as wheat, rice, and corn can be stored in elevators like these for months or years, but only if they are kept dry. Grains are never harvested and put into storage unless they are already extremely dry. Even so, the very slow respiration of each kernel produces a tiny bit of water, and if it accumulates, fungi can grow and spoil the grain. The elevators have large fans that blow dry air through the grain. Figure B11, 4b. The dead zone in the Gulf of Mexico is caused by the rapid growth and respiration of bacteria, which are nourished by fertilizers that pollute the Mississippi River and its tributaries. Wine. Wines are fermented fruit juices that are rich in sugars. When the word wine is used by itself, it refers to fermented grapes of the species Vitis vinifera. If other fruits are used, then they are named, such as elderberry wine or peach wine. As anyone who has shopped for or selected wine knows, there seems to be an endless variety of wines, such as Chardonnay, Pinot Noir, Cabernet Sauvignon, and Merlot. It may seem that these represent numerous species of grape, but actually all are varieties of just one, V. vinifera. Wine was first produced about 6000 BCE, and since then farmers have selected numerous varieties, each with its own special flavors, aromas, and colors, but always cultivated from V. vinifera. This is a little surprising because there are 60 species of grape in the genus Vitis. These others are typically used for jams, jellies, juices, raisins, or for eating fresh, but they are not used for wine. Grapes are harvested when they reach proper maturity and their sugar content is suitable. Typically, 20% to 30% of the grape's weight is sugar when harvested. They are washed and then crushed to obtain the juice. For white wines, the skins are removed, but for red or rose wines, the skins are mixed in with the juice. The skins not only provide the color of red wine, they produce various flavors and aromas. The chemical resveratrol is produced when yeasts attack the skins. There are claims that resveratrol lowers blood pressure, prolongs life, and has anti-cancer activity, but none of these has been proven definitively in humans. Cerevisiae is added to the juice, and the mixture is cooled slightly. White wines are fermented at 10 to 15 degrees Celsius and red wines slightly warmer at 25 to 30 degrees Celsius. Fermentation vats have valves that allow carbon dioxide to escape without allowing oxygen to enter. If oxygen were to enter the tank, certain bacteria would convert the ethanol to acetic acid, which would turn the wine into vinegar. After about 8 to 10 days, the liquid is removed from the skins and allowed to continue fermenting for up to 1 month. During fermentation, dead yeast cells